Hi everyone, this is lecture 4 of week 6. We're going to be continuing our discussion of memory, and now we're going to be focusing on the biology and development of memory. So I want to begin by telling you about a study. In the 1920s, psychologist Carl Lashley tried to locate what he called the engram, or the physical traces of memory in the brain. Excuse me. Or where do memories actually live in the brain? In an effort to discover where memories were stored, Lashley taught rats how to run various mazes. He then lesioned or damaged different parts of the brain to see if those rats forgot how to complete the maze. So, for example, he might lesion the amygdala and then put them back in the maze and see what happens. Lashley found no engram or evidence of an engram, but he did learn two important things. First, the more brain tissue he removed, the worse the rats performed. And second, no matter where he removed the brain tissue from, the rats retained at least some memory of the maze. These findings led Lashley to conclude that memory isn't located in just one single place. Memories of different types of experiences are stored in different brain regions. Long-term potentiation is the gradual strengthening of the connections among neurons from repetitive stimulation over time. This results in an increased release of glutamate. Most neuroscientists agree that long-term potentiation plays a key role in learning, and that the hippocampus plays a key role in forming lasting memories. But alas, there is no engram. Memories are diffusely stored throughout the brain. So where are memories stored? Well, we know that the hippocampus is critical, but learned information is not stored permanently in the hippocampus itself. As we've discussed, memories distribute themselves among different areas throughout the cortex. But I do want to talk a little bit about amnesia, or the loss of information. Explicit and implicit memories are governed by different brain systems, and evidence from this comes from amnesia studies. There's two common types. There's retrograde, or loss of memories from the past, and anterograde, or the loss of ability to make new memories. There's a third option that's not really based in any sort of scientific literature, but popular movies love it, and this is generalized amnesia, or the idea of someone losing their entire memory from before some sort of traumatic event. However, this is very rare. We hardly ever see cases where someone lost their entire memory. It's equally rare for the sudden recovery of memory as portrayed in movies. Memory recovery from amnesia tends to occur gradually, if at all. So one popular movie that shows an example of amnesia is Anastasia. So it's the story of a young girl, presumably the Grand Duchess of Russia, who wakes up one morning and, there's a traumatic experience in the movie, and after that she doesn't have any of her earlier memories. She can make new memories, so she is an example of retrograde amnesia. As is Dory, from Finding Nemo. Dory's actually an example of anterograde amnesia because she doesn't have the ability to make new memories. For those of you who have seen Finding Nemo, Dory is a character who forgets things as soon as they happen. So I want to show you this video now. There was a man who had a lobotomy to alleviate seizures and had part of his hippocampus removed. So that did help with the seizures, but it also had a really unfortunate side effect of damaging his memory. So let's take a look at this video. Ooh, excuse me.
1953, a desperate young man agreed to undergo an experimental procedure to remove a part of his brain in an attempt to cure his seizures. Henry Molaison had begun experiencing seizures as a child following a bicycle accident where he cracked his skull. At 27 years old, his seizures were so severe and uncontrollable that he was unable to lead a normal life. His surgeon, William Scoville, was a well-known lobotomist at the time. He had spent his career performing lobotomies on psychotic patients and noted that in some of those patients, the lobotomy had the unintended consequence of relieving their seizures. And so on September 1st, 1953, William Scoville removed a thumb-sized section from both sides of Henry's brain. The majority of the medial temporal lobe was removed, including the hippocampus, amygdala, and entorhinal cortex. The surgery did alleviate some of Henry's seizures, but it also left him unable to form any new memories, eventually becoming known in the medical literature as patient HM. Our understanding of memory at the time was rudimentary. Memory was thought to be distributed throughout the entire brain and tied to other brain functions, such as perception and intellect. But the surgery didn't affect Henry's perception, nor did it dim his intellect. In fact, he scored higher on IQ tests following his surgery. Similarly, his language and motor skills were entirely preserved. It seemed the only thing he had lost was the ability to form new memories. 2 years later, in 1955, Henry Molaison was visited by a memory researcher from McGill University, Brenda Milner, who then spent the next 3 decades of her career testing and studying Henry's brain, eventually passing him on to her former student Suzanne Corkins. Though Henry was studied extensively by both Brenda Milner and Suzanne Corkins, it was Brenda Milner's early observations that taught us most of what we know about memory. Milner found Henry to be the ideal study subject. He was highly intelligent, well-mannered, and could sit through hours and hours of testing without ever getting bored or complaining, always eager to cooperate. It was in those early years that we were able to conclude that memory, far from being distributed throughout the entire brain, was limited to specific brain structures, namely the hippocampus and adjacent perirhinal, entorhinal, and parahippocampal cortices. Together, they are now referred to as the medial temporal lobe memory system. But Henry's memory loss was an anterograde amnesia, meaning that he was unable to create any new memories while still being able to recall long-term memories from before the surgery. This is because while the medial temporal lobe is responsible for creating new memories, once these memories have been consolidated over time becoming long-term memories, they are then moved to be stored outside the medial temporal lobe in other parts of the brain. While Henry was able to recall personal events from his childhood with remarkable clarity and able to recognize the faces of famous people who were in the news before his surgery, Brenda Milner found herself having to introduce herself for the very first time every single time she met him. Milner observed that despite Henry's inability to create any new memories or remember things, he was still able to acquire new motor skills. He was able to learn new complex skills such as mirror drawing and was able to retain those skills. This was the earliest demonstration that there existed multiple memory systems that were housed in different parts of the brain. Eventually, we came to understand memory as being either declarative, the ability to recall factual information, or non-declarative, an umbrella term that describes skill learning, habit learning, and emotional memory. Things like walking, cycling, and nail biting. We now know that non-declarative memory depends on the basal ganglia, amygdala, cerebellum, and neocortex. These structures were all preserved during Henry's brain surgery in 1953, thus allowing him to acquire new skills and habits. You probably noticed that throughout the
Anyone know what kind of research design HM would be? You think back to week 2 when we talked about research methods. This would be an example of a case study because it's only one participant. So it's difficult for us to make sweeping generalizations, but does give us a good example of, if we were to do this procedure to the hippocampus, what does it do to memory? And lets us infer or make some sort of overarching ideas about what we think, where we think memory lives.
This video, I refer to Henry as simply Henry.
When we talk about emotional memory or memories with a significant emotional aspect, the amygdala and hippocampus play a huge role by interacting to give us emotional memories. The amygdala helps us to recall the emotions that are associated with fear-provoking events, and the hippocampus helps us to recall the events themselves. There's some different examples that differentiate the roles of each of these structures. Does anyone watch the show Outlander? It's a bit of a trigger warning in season 1, but there's a really disturbing episode at the end of the season where a character is exposed to the scent of lavender while being violently raped. Later, they show how the smell of lavender triggers memories of the traumatic experience for this character, showing how different parts of the brain work together to influence recall of memory. You can sort of think of it as a form of classical conditioning. The smell might be paired with the memory to evoke a fear response. This can also work in the positive direction. The smell of sunscreen might trigger pleasant emotions of fun days at the beach with your family. Unfortunately, things like age can impact our memories. After about age 65, humans begin to experience memory problems from some general deterioration in the brain. Although scientists, although scientists disagree about how much memory loss is normal in the aging process. We know that some is, but the exact question of how much is a little bit less clear. One disorder with more than average memory loss is dementia, which is often severe memory loss. Alzheimer's disease is the most common cause of dementia, accounting for 50 to 60% of cases. Another cause of dementia is brain damage that can occur from multiple strokes. Although dementia and Alzheimer's are frequently thought to be the same, they're actually different. Often you can't tell if someone has Alzheimer's without conducting a brain biopsy, which we'll talk about in a minute. Cognitive impairments are really common in Alzheimer's, and they relate to memory and language, which corresponds to patterns of cortical loss in these patients. So people with Alzheimer's often forget more recent events and memories first. So they might forget the names of their grandchildren, but still remember the names of their children. They often also experience disorientation regarding their current location or other information. Alzheimer's brains contain many senile plaques and neurofibrillary tangles that contribute to the loss of synapses and the death of cells in the hippocampus and cerebral cortex. Unfortunately, to date, there's no treatment that's effective in halting or reversing the effects of Alzheimer's. So I just want to show you a quick example of some of the cortical loss that's associated with Alzheimer's. So on the left, you see a normal brain, a typical brain. And on the right, you see the brain of someone with Alzheimer's. So as I hope you see, the brain on the right is smaller. It's missing quite a bit. So there's severe ventricle and cortical loss, including some of the areas that are associated with language and memory. We don't need to dive too deeply into the nitty-gritty here. I just want to give you a bit of an overview. And I want to show you a video that gives you some good detail as well.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss Alzheimer's disease. Alzheimer's disease is the most common form of neurodegenerative disease, meaning it is characterized by the degeneration and death of neurons. It is classified as a type of dementia, a term that refers to a category of brain disorders that involve memory loss and cognitive impairment. Alzheimer's most often affects adults over the age of 65. The causes of the disease are not well understood, and genetics and environmental factors are thought to be involved. Alzheimer's disease is associated with neuronal death throughout the brain, which can be extensive enough that regions of the brain appear atrophied or shrunken compared to a healthy brain. A hallmark sign of Alzheimer's disease is the formation of clusters of misfolded proteins both inside and outside of neurons. One of these proteins, amyloid beta protein, is found in the extracellular space around neurons in a healthy brain. During Alzheimer's, however, misfolded forms of amyloid beta clump together in deposits called amyloid plaques. Another protein called tau protein, which is normally found inside neurons and involved in maintaining neuronal structure, is also found in a misfolded state in Alzheimer's. It accumulates inside neurons in bundles called neurofibrillary tangles. Although amyloid plaques and neurofibrillary tangles are hallmark signs of Alzheimer's disease, it is unclear if they contribute to the neurodegeneration or are a part of the brain's response to it. The most common treatments for Alzheimer's disease involve drugs that inhibit the activity of acetylcholinesterase, an enzyme that breaks down the neurotransmitter acetylcholine. The drugs, called acetylcholinesterase inhibitors, increase levels of acetylcholine, which is thought to promote healthy cognition and memory. The effects of these treatments are modest, however, and they do not stop neurodegeneration in Alzheimer's. Thus, they are not a cure for the disease. Welcome to
So now let's take a look at memory across time. Memory changes as we age, but there's considerable continuity that remains across development. Memory span and the ability to use strategies increase dramatically across infancy, toddlerhood, preschool age, and elementary school years, and children's memories become increasingly sophisticated as they get older. Their memory span increases with age as well, which may be due to more strategies that they learn in school and from exposure to other same age peers, as well as their conceptual understanding increasing and their ability to chunk related items and store memories in more meaningful ways. We also develop meta-memory skills as we age. These are the knowledge of one's own memory abilities and limitations. So they might understand more about what they can remember and what they can't. Ever try to explain something to a young child? You have to be very simplistic and clear because their memory systems are super underdeveloped. But an older child can remember more and is more responsible and able to complete and remember more complex tasks. This is due to how memory develops as we age. I'm not going to go too far into details, but suffice it to say that our memories get better and better as we progress from childhood to adulthood, but then taper off after about age 65, as we just discussed. Our long-term memory is finite. After a point, it fills up and we can't retain anything else. So how do we make room for new memories? We do so through a process called memory pruning, which is your brain's version of restarting and clearing up the memory banks. Some scientists aren't really sure how memories work. They're not positive about how your brain does it, but it basically tosses out old questionable memories to make room for new ones. What they're saying is that memories fight each other when you're trying to remember them, so the brain automatically decides which ones are not worth keeping and tosses them in the trash. Without that process, you'd probably become so overwhelmed by the sheer quantity of stuff rattling around in your brain that you couldn't possibly remember all the things you want to. I don't know why this isn't working. Let's take 1 second and see if we can get it. If not, then not. It's just a clip from Inside Out that I thought was fun. Come on. No, it's not going to work. That's okay. You can check it out on the slides themselves. So up next, we're going to talk about false memories, which there's some really interesting literature. I think you'll find that really exciting. So I will talk to you then.